Welcome to the deep dive. Today we're exploring something your body does almost every day, often without you even noticing until, well, you really notice it. Acute inflammation.
That's right. When you get that cut or maybe bump your knee and suddenly it's red, hot, swollen, and hurts. That whole package.
Exactly. It feels like chaos, but it's actually this incredibly coordinated response, like an emergency broadcast system kicking in.
Precisely. And behind the scenes, pulling all the strings, are these chemical signals. That's our focus today. The mediators of inflammation. These are the molecules that start, manage, and eventually stop the whole process.
Yeah, and when you first look at the list of them, cytokines, chemokines, complement, it just seems like, wow, an alphabet soup of biology. It can feel a bit overwhelming.
It really can. There's so many players, but I think the best way to make sense of it, the key really, is to group them by where they come from. Because their origin story dictates how fast they act and what they do.
Okay, origin story. So what are the main camps?
Fundamentally, there are two big categories. First up, cell-derived mediators.
Meaning they come from cells already on the scene.
Exactly. Think of your local security team, the sentinel cells. These are macrophages, mast cells, dendritic cells, always patrolling the tissues, especially near blood vessels.
Ready for trouble.
Ready for trouble. And these cell-derived signals work in two ways. Some are like prepackaged, think of histamine. It's stored in little granules inside mast cells, ready to be dumped out instantly upon injury. Degranulation.
Like pulling a fire alarm, instant signal.
Bang. Instant signal. Then there's the second type, like prostaglandins and leukotrienes. These aren't pre-made. They have to be synthesized de novo, basically built from scratch when the cell gets the right stimulus.
So that takes a little longer, I guess.
A little bit longer, yeah. But it allows for a more tailored response, maybe a more sustained one, compared to the immediate histamine blast.
Got it. Pre-made alarms and custom-built response teams. What's the second big category?
The second camp is the plasma-derived mediators.
Yeah.
These are fascinating. They're mostly proteins made by the liver, and they just circulate constantly in your blood plasma.
Just floating around.
Yeah, but crucially, they're inactive precursors. Think of them like unassembled components or maybe dormant landmines waiting for a very specific trigger.
Okay, so they need activation, like the complement system, or kinins.
Like exactly those. They require some kind of enzymatic cleavage, basically being cut in a very precise way to become active. And the cool thing, or maybe the dangerous thing, is that activating one often triggers a whole chain reaction, a cascade.
A cascade. That sounds like it could get out of hand quickly.
It could. Which brings us to some really key principles about how these mediators work. Why doesn't this powerful system just, burn the house down every time?
Right. What are the built-in controls?
Well, the source material highlights three really important ones. First, they're generally produced only when needed. There has to be a specific trigger, microbes, damaged cells, injury. No trigger, no mediators.
It's stimulus-driven, not just always on.
Exactly. Second, and this is vital for control, they are typically very short-lived. They decay rapidly, or they get broken down by enzymes or just mopped up. Their signal is brief.
So they don't hang around causing trouble longer than necessary. A short half-life is the safety switch.
It really is.
Yeah.
And third, this idea of cascades and amplification. One mediator can trigger the release or activation of many others. Think TNF stimulating IL-1 or complement activating histamine release.
That creates redundancy, right? If one signal gets blocked, others can step in.
Absolutely. It makes the whole system robust. Hard to completely shut down if there's a real threat. Okay, so should we dive into the specific groups now, starting with the fastest responders?
Let's do it. The vasoactive amines, the first alarm.
And the superstar here is undoubtedly histamine, often called the master switch. It's primarily stored in those granules within mast cells, which, remember, hang out right next to blood vessels. Perfect location.
Basophils and platelets have some too, right? But mast cells are the main source.
Mast cells are the big ones for that immediate tissue response, yeah. And the speed of release is key.
What actually makes those mast cells degranulate? What flips the switch?
Well, there are a few main triggers. Simple physical stuff, like direct trauma, heat, cold, anything that damages the cell membrane can do it.
Makes sense. Injury equals histamine.
Then there's the immune trigger, the one people know from allergies. An antigen, like pollen, binds to IgE antibodies already sitting on the mast cell surface. That triggers a signal. Calcium rushes in, and boom, histamine release.
Which explains why peanut allergies or bee stings can cause reactions so incredibly fast. Seconds, sometimes.
Exactly that fast. And the third trigger loops back to those plasma proteins we mentioned. Specific bits of the complement system, C3a and C5a, are potent signals. They float around, find mast cells, bind to them, and tell them to release histamine.
Wow, so the complement system directly amplifies the histamine effect. A feedback loop.
A powerful feedback loop. So histamine floods out. What does it actually do in those first few moments?
That's where we see the classic signs starting, right? The redness and swelling.
Precisely. Histamine has two main, very rapid but transient effects. First, it causes arterioles, the small arteries leading into capillary beds, to dilate. More blood flow.
That's the heat, calor, and the redness.
Ruber. Yep. Second, it acts on the venules, the small veins leaving the capillary beds. It makes the endothelial cells lining those venules contract slightly, pulling apart.
Creating gaps between the cells.
Exactly. And those gaps dramatically increase the permeability of the vessel wall. Protein-rich fluid, and even some cells, start leaking out from the blood into the surrounding tissue.
And that's the swelling, the edema or tumor, the fluid buildup.
That's the one. It happens almost immediately, but the histamine effect itself only lasts, maybe 15, 30 minutes. It kicks things off dramatically, but other mediators have to sustain it.
Which is why antihistamine drugs work so well for allergies, right? They block the histamine receptors.
Exactly. They're usually H1 receptor antagonists. They sit on the receptor, block histamine from binding, and prevent that initial vasodilation and permeability surge. You're essentially jamming the master switch.
Makes sense. Now, the other vasoactive albumin mentioned is serotonin. What's its role? Is it as important?
Serotonin, or 5-HT, is mostly known as a neurotransmitter, of course. But platelets store quite a bit of it. When platelets aggregate during clotting, they release it.
And what does it do in inflammation?
Its effects are a bit more complex, sometimes vasoconstriction, sometimes contributing to permeability. But overall, its role in the classic acute inflammatory response seems less central compared to histamine, maybe more of a supporting actor.
Okay, so histamine is the big initial bang. What comes next? We need something to sustain and amplify that response locally.
That brings us beautifully to our second major group, the lipid messengers, particularly the eicosanoids. If histamine was the alarm bell, these guys are like the local field commanders setting up the perimeter and calling in reinforcements.
And these all come from a common source molecule hidden in our cell membranes.
That's right. Arachidonic acid, or AA. It's a 20-carbon fatty acid, usually esterified, tucked away in membrane phospholipids. It's just sitting there until the cell gets activated.
Activated how? What shakes the AA loose?
The key enzyme is phospholipase A2. Various stimuli, physical damage, chemical signals, even other mediators like C5a from complement can switch on phospholipase A2.
And it acts like molecular scissors.
Pretty much. It snips the AA molecule free from the membrane phospholipid. Once AA is liberated, it's immediately grabbed by other enzymes and converted into these super potent signaling molecules, the eicosanoids.
Which include prostaglandins, leukotrienes.
And lipoxins, exactly.
Yeah.
And these eicosanoids bind to specific receptors, usually G protein-coupled receptors, on many different cell types, driving almost every aspect of inflammation.
Pain, fever, vascular changes, leukocyte movement.
You name it, eicosanoids are probably involved. And this AA pathway splits into two main branches, which is really important pharmacologically. Let's start with the cyclooxygenase, COX pathway, which makes prostaglandins, PGs.
COX-1 and COX-2. This is where understanding the difference is crucial for things like NSAIDs.
Absolutely critical. So COX-1 is what we call conspicutive. It's basically always on at some level in those tissues. It does essential housekeeping jobs.
Like protecting your stomach lining.
Exactly. It helps produce prostaglandins that stimulate protective mucus in the stomach, maintains kidney function and blood flow, helps regulate platelet function, normal everyday stuff.
The good COX, you might say?
Well, the essential COX for normal physiology, yeah. COX-2, on the other hand, is inducible. It's normally present at very low levels, but it gets massively upregulated in cells involved in inflammation macrophages, endothelial cells, fibroblasts when they're hit with stimuli like TNF or IL-1.
So EOX2 is the inflammatory COX switched on during battle.
That's the idea. And this distinction drove the whole effort to create COX-2-specific inhibitors. But let's look at the prostaglandins themselves first. There's a really important balance right within the blood vessel.
You mean between clotting and anti-clotting signals?
Precisely. Platelets primarily use COX-1 to make thromboxane A2, TXA2. TXA2 is a potent vasoconstrictor and strongly promotes platelet aggregation. It wants to form a clot.
Okay, stop the bleeding. Makes sense.
But the endothelial cells lining the blood vessel use COX-2 and COX-1 to make prostacyclin, PGI2. PGI2 does the exact opposite. It's a vasodilator and a powerful inhibitor of platelet aggregation. It wants to keep the blood flowing smoothly.
So a constant tug of war, platelets trying to clump and endothelium trying to keep things open.
A vital balance. And disrupting that balance, as we'll see later with COX2 inhibitors, can have serious consequences.
Okay, what about the prostaglandins that cause the obvious symptoms, like pain and swelling?
Several prostaglandins contribute to vasodilation and increased permeability, working alongside or after histamine. PGD2 is a major one made by mast cells. PGE2 is also a big player in vasodilation and edema.
And PGE2 is the main culprit for pain and fever, right?
It's strongly implicated, yes. For pain, PGE2 doesn't usually cause it directly, but it makes nerve endings hypersensitive to other painful stimuli like bradykinin or even just pressure. It lowers the pain threshold, hyperalgesic.
Makes everything hurt more. Ouch.
Exactly. And for fever, PGE2 acts centrally in the hypothalamus of the brain. Inflammatory signals like IL-1 trigger PGE2 production there, which essentially resets the body's thermostat upwards.
Which is why drugs like ibuprofen or aspirin, which block both COX1 and COX2, are good at reducing both pain and fever if they stop PGE2 production.
Correct. They hit the molecule responsible for both symptoms.
All right, let's switch to the other major branch of the AA pathway, the lipoxygenase pathway making leukotrienes, LTs.
Right. The key enzyme here is 5-lipoxygenase, which is found in abundance in neutrophils, those early responder white blood cells.
Yeah. And what do the leukotrienes do?
One major product is leukotriene B4, LTB4. This molecule is basically a super powerful chemoattractant specifically for neutrophils. It creates a chemical trail, leading them right to the site of injury or infection. It also activates them once they arrive.
Like a chemical flare guiding the troops and getting them ready to fight.
Excellent analogy. Then you have the other group, the cysteinoleukotrienes, LTC4, LTD4, and LTE4. They're called that because they have a cysteine amino acid attached.
And these are the power brokers you mentioned. What makes them so potent?
They cause intense vasoconstriction in some areas, but their really big impact is causing profound bronchospasm tightening of the airways. This makes them absolutely central players in asthma.
More so than histamine for breathing problems.
Oh, yes. They are also much more potent than histamine at increasing vascular permeability. Their effects are slower to develop than histamines, but much more sustained. Think hours rather than minutes. They really drive that prolonged leaky vessel effect.
So histamine for the initial shock, leukotrienes for the sustained siege.
Kinda, yeah. Now here's the elegant part. The same overall pathway that generates these pro-inflammatory molecules also generates signals to stop the inflammation. The lipoxins, LXs.
The brakes on the system. How do they work?
Lipoxins actively suppress inflammation. Their main job seems to be inhibiting neutrophil recruitment and adhesion. Once enough neutrophils have arrived, lipoxins essentially tell incoming ones, okay, stand down, we've got this. They promote resolution.
That's crucial. How are they made? Is it simple?
Actually, no, it's quite sophisticated. Making lipoxins requires a process called transcellular biosynthesis. It needs cooperation between two different cell types.
Really? Like which ones?
Typically, neutrophils start the process. They take AA and partially process it using their lipoxygenase. Then they pass this intermediate molecule over to platelets that might be adhering nearby.
And the platelets finish the job.
Exactly. Platelets have the final enzyme needed to convert that intermediate into the active lipoxin. It's this little molecular handshake ensuring that the stop signal is generated right where the action, including platelet aggregation, is happening.
That's incredibly elegant. A built-in off switch requiring teamwork. Okay, we've covered the local alarms and amplifiers. Let's move up the command chain. Cytokines and chemokines, the recruiters and systemic controllers.
Absolutely. We have to start with the two heavyweights, TNF, tumor necrosis factor, and IL-1, interleukin-1. These are small proteins, mostly churned out by activated macrophages and dendritic cells, those sentinel cells again.
What triggers their production?
Direct contact with microbial products, like bacterial endotoxin or signals from damaged host cells. They are a primary response to danger signals.
And what do they do locally, right at the site of inflammation?
Their key local job is endothelial activation. They dramatically change the behavior of the endothelial cells lining the small blood vessels.
Making them sticky, like you said.
Exactly. TNF and IL-1 cause the endothelial cells to express adhesion molecules on their surface. First, things called selectins, E-selectin, P-selectin, which act like weak speed bumps, causing passing leukocytes to slow down and start rolling along the vessel wall.
So they're not just whizzing by anymore.
Right. Then, TNF and IL-1 induce the expression of ligands for integrins molecules like ICAM-1 and VCM-1. Integrins are adhesion molecules on the leukocytes themselves.
Okay, so the endothelium puts up the docking sites.
And then other signals, especially chemokines, cause the integrins on the rolling leukocyte to switch from a low-affinity state to a high-affinity state. They suddenly drip tightly onto ICAM-1 or VCM-1.
Firm adhesion. The cell stops rolling and sticks fast.
Sticks fast, flattens out, and then squeezes between the endothelial cells to get into the tissue. TNF and IL-1 orchestrate this whole recruitment process. They also stimulate the endothelial cells and macrophages to produce more mediators, like chemokines and even more IL-1, amplifying the whole response.
A positive feedback loop locally, but they also have effects far beyond the local site, right? The systemic stuff.
Oh, big time. This is the systemic acute phase response. TNF and IL-1 travel through the bloodstream and act on distant organs. They hit the hypothalamus in the brain to cause fever.
Okay, fever. What else?
They stimulate the liver to rapidly produce a whole suite of acute phase proteins, like C-reactive protein, CRP, and fibrinogen, which we can measure in blood tests as markers of inflammation. They can cause lethargy, loss of appetite.
That general feeling of being sick.
That's largely TNF and IL-1 talking to your brain. And in severe infections or chronic inflammation, sustained high levels of TNF can cause a really serious problem called cachexia.
That's the wasting syndrome, losing muscle and fat.
Exactly. TNF essentially tells the body to break down its energy stores, muscle protein, fat reserves, leading to profound weight loss and weakness. It's a major issue in chronic diseases like cancer or long-term infections. Shows how a good mediator can turn bad if it sticks around too long.
A very fine line. Okay, let's drill down on the specific signals that guide the traffic, the chemokines.
Right, the chemoattractant cytokines. Think of them as the molecular GPS system for immune cells. They form a chemical concentration gradient, highest at the source of inflammation, and leukocytes follow that trail.
Like breadcrumbs leading them to the target.
Precisely. And they're classified based on their structure, specifically the arrangement of cysteine amino acids near the start of the protein.
You mentioned CXC and CC.
Those are the two major families. CXC chemokines, where the first two cysteines are separated by one other amino acid, the X, primarily act on neutrophils. The classic example is IL-8, now called CXCL8. It's a major recruiter of neutrophils to sites of bacterial infection.
Got it. Neutrophil callers. What about CC?
In CC chemokines, the first two cysteines are right next to each other. These tend to attract monocytes, eosinophils, basophils, and lymphocytes, more the mononuclear crowd.
So different chemokines call in different types of responders.
Generally, yes. For instance, MCP1, monocyte chemoattractant protein 1 or CCL2, is a big recruiter for monocytes, which mature into macrophages in the tissue. Eotaxin, CCL11, specifically calls in eosinophils, important in allergic reactions and parasitic infections.
It's like sending out specific invitations for the right kind of help.
Exactly. And remember, besides just attracting them, chemokines also play that crucial role in activating the integrins on the leukocytes, ensuring they stick firmly to the endothelium before migrating out.
So they guide and help them grab on. Do they have roles outside of inflammation?
Oh yes. They're also vital for normal immune cell trafficking and development, maintaining the architecture of lymphoid organs like lymph nodes and the spleen. They direct cells to the right zones even in peacetime.
And you mentioned that some viruses, like HIV, actually hijack these chemokine receptors.
It's a remarkable example of viral ingenuity, unfortunately. HIV uses the normal chemokine receptor CXCR4 and CCR5, which are supposed to bind CXC and CC chemokines respectively as co-receptors to actually get inside T cells and macrophages.
Wow. It uses the cell's own doorway.
It literally picks the lock, using the mechanism designed for immune cell communication. It highlights how fundamental these pathways are.
Are there any other key cytokines we should touch on quickly?
IL-6 is definitely important. It works alongside TNF and IL-1, especially in driving that systemic acute phase response from the liver, and maybe IL-17.
IL-17, what's its specialty?
IL-17 is mainly produced by a specific subset of T helper cells, Th17 cells, and is a really potent inducer of chemokine production by other cells. Its main downstream effect is massive recruitment of neutrophils and monocytes.
So it's like an amplifier for the recruitment signal.
A major amplifier. And we know it's important because people with genetic defects in the IL-17 pathway often suffer from something called cold abscesses.
Cold abscesses, what does that mean? They're not actually cold.
Well, they lack the intense heat and redness, the calor and rubor that you typically associate with a bacterial abscess. Because the massive neutrophil influx, which causes a lot of those signs, is impaired without strong IL-17 signaling. The infection is there, but the classic inflammatory fireworks are missing.
Fascinating. Shows how directly these mediators link to the symptoms we see. Okay, that covers the cell-derived signals pretty well. Let's move to the big guns circulating in the blood, the plasma protein systems, starting with the complement system.
Complement. A beautiful, complex, and sometimes terrifying cascade. It's a collection of over 20 proteins, mostly inactive precursors, circulating in the plasma. Its activation is like a molecular chain reaction.
And it all hinges on activating one key component.
The central event, the point of no return for complement, is the cleavage of C3. Everything leads to activating C3.
How does the C3 get activated? The sources mention 3 different ways to kick it off.
3 main pathways, all converging on C3. First, the classical pathway. This one bridges innate and adaptive immunity because it's typically triggered when antibodies, IgG or IgM, bind to an antigen, like on a bacterium. The C1 complex binds to these antibodies and starts the cascade.
So antibodies flag the target, complement attacks.
Basically, yes.
Right.
Second, the alternative pathway. This is pure innate immunity. It can be triggered directly by microbial surfaces themselves, things like endotoxin, LPS, on bacteria, or certain yeast cell walls. No antibodies needed. It's like a constant, low-level surveillance system that ramps up on foreign surfaces.
Always ready to go. And the third one?
The lectin pathway. This uses a molecule called mannose-binding lectin, MBL, which circulates in the plasma. MBL recognizes specific sugar patterns, mannose residues, that are common on microbes but not usually on human cells.
Yeah.
When MBL binds, it activates proteases that kick off the cascade, similar to the classical pathway, but antibody independent.
So antibodies, microbial surfaces, or microbial sugars can all light the fuse, and they all lead to?
They all lead to the generation of an enzyme complex called the C3 convertase. Its job is simple. Chop C3 into 2 pieces, C3A and C3B. And once that happens, the major functions of complement are unleashed.
Okay, what are those big functions? Function 1.
Inflammation. The smaller fragment, C3A, along with C5A, which is generated downstream when C5 is cleaved, are potent anaphylatoxins. We mentioned them earlier. They trigger mast cell degranulation, releasing histamine and causing vasodilation and increased permeability.
More swelling and redness.
Yep. And C5A is also one of the most powerful chemoattractants known in the body. It calls in neutrophils, monocytes, eosinophils, basophils, basically everyone to the site.
A major recruitment signal. Okay, function 2.
Opsonization and phagocytosis. The larger fragment, C3b, is the key here. It covalently attaches itself to the surface of the microbe or particle that triggered activation. It coats the target.
Opsonization means making tasty, right?
Essentially, yes. Phagocytic cells, like macrophages and neutrophils, have specific receptors on their surface that recognize and bind tightly to C3b. So coating a microbe with C3b is like putting little handles all over it, making it incredibly easy for phagocytes to grab on and engulf it.
Marking it for destruction. Very efficient. And function 3.
Direct cell lysis. This involves the later complement components from C5b down to C9. They assemble together on the target cell surface to form the membrane attack complex, or MA.
The MAC attack, what does it do?
It literally forms a pore, a channel, right through the microbial cell membrane. It's like punching holes in it. Water rushes in, essential ions leak out, and the cell swells and bursts. Lysis.
Ouch. Effective, but sounds dangerous if it happens to your own cells.
Extremely dangerous, which is why our own cells have multiple protective mechanisms. There are regulatory proteins in the plasma, like C1 inhibitor, C1-INH, that shut down early steps. And crucially, our own cells have proteins embedded in their membranes, like decay accelerating factor, DAF, and CD59, that actively disrupt C3 convertase formation or block the MAC assembly right on the cell surface.
A built-in self-defense shield against friendly fire. Smart.
Absolutely essential. Without it, complement would constantly damage our own tissues.
Okay, that's complement. What about the other plasma system, the kinins?
The kinin system is another cascade. It starts with inactive precursors called kinogens in the plasma. Enzymes called kallikreins cleave these kinogens to release active vasoactive peptides.
And the main one we need to know is bradykinin. That's the star player.
What does bradykinin do? Similar to histamine?
It shares some effects, yes. Bradykinin causes vasodilation, increases vascular permeability, contributing to swelling, and causes contraction of smooth muscle. But its most famous or infamous effect is pain.
So bradykinin is a major pain mediator, a very potent one. It directly stimulates sensory nerve endings, contributing significantly to the tenderness and pain associated with acute inflammation. If something hurts when it's inflamed, bradykinin is probably involved.
Like histamine, is it short-lived?
Yes, thankfully.
Yeah.
It's rapidly broken down by enzymes called kininases in the plasma and tissues, so Its effects are usually quite transient unless the system is continuously activated.
Okay, wow. We've covered a huge amount of ground. Amines, lipids, cytokines, chemokines, complement, kinins. It's this incredibly complex interwoven network.
It really is. Redundant, powerful, tightly regulated
Mm-hmm.
most of the time. It's the chemical language of immediate defense.
Let's shift gears now and talk about how understanding all this chemistry translates into medicine. How do we try to control this cascade? This brings us to updates and advancements.
Yeah, this is where the basic science meets the clinic. Knowing the pathways allows for target therapies. But it's always that double-edged sword we talked about. Dampen inflammation, and you might compromise necessary defense.
Let's start with the eicosanoids again. The COX inhibitors. We touched on NSAs blocking both COX-1 and COX-2.
Right. Drugs like aspirin, ibuprofen, naproxen, they're non-selective. They reduce pain and fever by blocking COX-2-derived PGE2, which is great. But they also block the protective COX-1 in the gut and kidneys.
Leading to side effects like stomach ulcers or kidney problems in some people?
Exactly. Which led to the development of selective COX-2 inhibitors, the COXIBs. The idea was brilliant. Block only the inflammatory COX-2, spare the protective COX-1.
And they worked for inflammation and pain with fewer stomach issues. But then came the COX-2 dilemma.
The cardiovascular risk, yes. It turned out that selectively blocking COX-2 could tilt that delicate balance we discussed earlier, the one between platelet TXA2, made by COX-1, proclotting, and endothelial PGI2, made mainly by TOX2, anti-clotting.
So by knocking out PGI2 production while leaving TXA2 untouched.
You shift the balance towards a prothrombotic state, more platelet aggregation, more vasoconstriction. In people already at risk, this increased the likelihood of heart attacks and strokes.
A major reminder that you can't always target just one piece of a complex system without consequences.
A very stark reminder. It really highlighted the importance of that COX-1-COX-2 balance in vascular health.
What about targeting the other side, the leukotrienes? Has that been more straightforward?
For certain conditions, yes. Especially asthma. Remember, cysteine leukotrienes cause bronchospasm and increase permeability in the airways.
So drugs that block them should help breathing.
And they do. There are two main classes. 5-lipoxygenase inhibitors, like zeluton, stop the synthesis of all leukotrienes, including LTB4, and leukotriene receptor antagonists, like montelukast, efgartigimod, specifically block the cis-LT1 receptor, where LTC4, D4, and E4 act. Both are effective therapies for asthma management.
Okay, moving to the cytokines. Cytokine antagonism, especially targeting TNF, seems like a huge success story for chronic diseases.
It has been revolutionary. Anti-TNF therapies, drugs like infliximab, adalimumab, etanercept, have transformed the treatment of rheumatoid arthritis, psoriasis, Crohn's disease, conditions driven by chronic TNF overproduction. They can dramatically reduce inflammation and prevent tissue damage.
But there's a significant downside, right? The infection risk.
Yes, that's the trade-off. TNF is crucial for controlling certain intracellular pathogens, particularly Mycobacterium tuberculosis, the bug that causes TB. By blocking TNF, you impair the ability of macrophages to contain these infections.
So patients need to be screened for latent TB before starting these drugs.
Absolutely essential. There's an increased risk of reactivating latent TB or acquiring other opportunistic infections. It underscores TNF's vital role in normal host defense.
Are we seeing similar success targeting other cytokines now?
Yes, the field is advancing. Antagonists against IL-6 receptors, like tocilizumab, are very effective in rheumatoid arthritis and other conditions. And drugs targeting IL-17 or its receptor are proving highly effective for psoriasis and psoriatic arthritis, fitting with IL-17's role in driving neutrophil recruitment to the skin and joints.
So more specific targeting is becoming possible. Now, what about the really cutting-edge idea? Not just blocking inflammation, but actively promoting its resolution. Active termination strategies.
This is a super exciting area. We used to think inflammation just sort of faded away when the stimulus was gone. But now we know it's an active, highly programmed process of resolution.
Driven by specific stop signals, like the lipoxins we talked about.
Exactly.
Yeah.
Lipoxins are key players in telling neutrophils to stop coming in. But there are others too. Anti-inflammatory cytokines like TGF-beta and IL-10 are produced later in the response, and they actively suppress macrophage activation and promote tissue repair.
Switching the whole program from attack to rebuild.
That's the idea. And this ties into another fascinating area, the resolvin connection linked to diet.
Resolvins. What are they? Related to lipids again.
Yes, they were derived from omega-3 polyunsaturated fatty acids, PUFAs, the kind you find in fish oil, EPA and DHA. It turns out that during inflammation, these omega-3s can be converted by enzymes, including lipoxygenases, into specific families of molecules called resolvins and protectants.
And these are anti-inflammatory.
Actively pro-resolution is maybe a better way to put it. They do things like stop neutrophil infiltration, stimulate macrophages to clean up dead cells and debris, a process called efferocytosis, and promote tissue regeneration.
So eating more fish oil could potentially shift your body's response towards producing more of these resolution signals instead of just pro-inflammatory ones.
That's the hope and the focus of a lot of research. By changing the lipid building blocks available, you might be able to tune the system to resolve inflammation more efficiently. It's about leveraging the body's own off switches.
Instead of just blocking the on switches, that's a really different way of thinking.
It is, moving from anti-inflammation to pro-resolution.
This has been incredibly illuminating. We've journeyed from the first alarm signals to the complex cascades, and even how we're learning to actively turn them off.
So just to quickly recap for you listening, we have these four main groups. The rapid vasoactive amines like histamine, the local lipid messengers, prostaglandins, leukotrienes, and the resolving lipoxins, the systemic cytokines and chemokines like TNF, IL-1, IL-6, IL-8, orchestrating the bigger picture, and the powerful plasma protein systems like complement and kinins bringing amplification and specific functions like lysis and pain.
And we can see how they create the symptoms. Histamine, PGs causing vasodilation, that's the heat and redness. Histamine, leukotrienes, bradykinin increasing permeability, that's the swelling. And PGs and bradykinin driving the pain. It all connects back.
It really does. But remember, the cells that execute the cleanup, the leukocytes guided by all these mediators, they work by releasing some pretty toxic stuff, reactive oxygen species, enzymes designed to kill microbes, but capable of damaging host tissue too.
Which leads us to that final, maybe slightly unsettling thought. When this powerful defense system, directed by this intricate cascade, goes awry, maybe it doesn't shut off properly in chronic disease, or it mistakenly attacks self in autoimmune conditions.
Yeah.
Is the biggest long-term danger actually the original trigger, or does the defense response itself become the primary threat to your own body?
That's the profound question, isn't it? When defense turns into self-destruction, it highlights just how critical the balance and regulation of these inflammatory mediators truly are. The very system designed to protect us holds the potential for immense harm if its controls fail.
A crucial balance indeed, something for all of us to think about. Thank you for guiding us through this deep dive today.
My pleasure. It's a fascinating system.